Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and our live session has already started. So if you have any queries, you can ask me the questions. Uh, Yasha, I think what you are asking me is out of the syllabus of this course anyway, but since you asked me, I will tell you that the recent research areas, which are upcoming areas, are the nanobiotechnology and the tissue engineering and stem cell biology, and mostly some more areas are coming up like mechanobiology and those and artificial intelligence and those kind of thing where you can actually work on the interface, so biology and chemistry, or biology or computer science and biology and mechanical engineering and so on. So now things are, such areas are becoming more and more interdisciplinary rather than the one particular focused area, and that's why it is important for you to have a better idea about your core course, but you should also have a general idea about all other subjects as well. And that's why it is important to take the several MOOC courses so that you have a very good idea about the different fields, so that it will help you in terms of the pursuing the research as well as the refining the results as well. Anvesha, I think you're asking something which is beyond this course, so I think I would prefer to have the questions what are related to this course only. I don't think we have ever discussed about the CRISPR-Cas and all those kinds of things. So I probably will prefer to give you the answer only on this course itself. Uh, Kami-sama is asking me how this course could be useful. Uh, okay, so I think this course is going to give you a general biology flavor, and in addition to that, I have tried to cover many things related to genetic engineering and then the downstream processings. So this course is very useful in terms of if you are would like to pursue the research in the area of biotechnology, and especially if you are interested to pursue something which is related to cloning, overexpression, and purification kind of research. So this, based on this course, you could be very confident about pursuing the research in the area of X-ray crystallography, in the area of molecular biology, in the area of cell biology, and so on. And apart from that, this course, the content, the way I have covered the content is very useful in terms of preparing even for the some of the competitive exams as well. For example, you can easily cover a very large chunk of the syllabus of the GATE also is being covered. So if you take the GATE biotechnology, I have tried to cover the whatever is being within the biotechnology. Apart from this course, if you study about the biochemical engineering and process biochemistry, and a little bit about the immunology, more or less, I think the course is very much covering the whole content related to the GATE syllabus of biotechnology. So I think Dr. Gautam is asking me whether this course is useful for CAS in university. So I'm very much unaware of what is the CAS actually. So if you elaborate, I think I will tell you what is CAS. As such, I really don't know what is CAS actually. And as far as the what Kami-sama is asking me, that she wants to pursue the how I should apply for PhD and job abroad. So I think if you want to do PhD, first you have to make a choice whether you want to do PhD from India or abroad, and then if you have already decided that you want to do PhD from abroad, what I will suggest is you have two way of pursuing it. Either you clear the some of the courses like GRE and TOEFL and all those kind of thing, and then you apply for the US and other countries, or I would suggest that you should actually take up some, if you are not interested to pursue the PhD from India, but then so you what you can do is you can take up some search positions and like some junior research positions or something like that, so that at least you have a good experience of handling the different types of reagents and performing some of the experiments. Because what I have realized is now in India the experimental part is pretty weak for the students. So when they, if you abruptly go without even being trained experimentally, then it would be difficult to pursue the research, and you will not be able to you will not be able to gain much, even if you go to any lab in Harvard or Stanford or anywhere, because you don't want to waste your precious time of your PhD time in learning the techniques. So, and technique part is pretty compromised, and people are not giving much weightage as far as the giving the lab experiments to the students. So better to work in a lab for 6 months or 8 months. During that period, you can prepare for a PhD and all the kinds of thing, and then you apply for the PhD outside, and that will actually make you more confident in pursuing the research related to biology. Uh, yeah. Kesha, I really don't know if any workshop is going on related to genetic engineering. Currently, there is no workshop anyway going on. Whatever is going on is a virtual mode, and virtually you cannot learn anything. So once the pandemic is over, probably there will be some opening in IITs and other places, and that time you can explore and get the idea about the workshops in different institutions. Uh, So, as the Dr. Gautam said, that CAS means Career Advancement Scheme. So, I think the both the courses like genetic engineering course or the experimental biotechnology course or most of the MOOCs courses are actually under the CAS program. So, if you take these courses, probably they will give you some weightage, and probably the institute will actually going to give some weightage to these courses. And if you are still having a doubt, probably you can ask the the related technical query to the NPTEL office or NPTEL team, both at IIT Guwahati or at the IIT Madras as well. So Mahek is also asking about the scope of this course, and I think when I was discussing about the questions from the Kami-sama, I have discussed what is the scope of this course. So probably I will suggest that uh, she can go through that content of the live sessions, and then uh, So this course is actually theoretical course. So I don't think uh, most of the biotechnology related companies like Biocon or uh, Jubilant Biosciences or those kind of companies where, and even like the Reddy's lab also, if they have a opening in the vaccine development team or somewhere where they are trying to just make the proteins and so something like that. So if those kind of opening is there, probably you can be able to apply to those companies, and you will definitely get, like for example, the Reliance Life Sciences and all those kinds of places where, so you cannot just directly get into the company and start working, but with this course, probably you will have a better idea about the experiments. You might have seen that in some of the lab sessions, some of the theoretical part, we have also introduced the lab sessions. So there we have shown you how to do the experiment as well. So that probably is very, that is the what best what we can do to to give you a real feeling how to do the things. And even in subsequently to this also, when we are going to interact in the next live sessions also, you will see that we'll going to see more lab experiments within the course also, and so on. So I think that the purpose of these lab sessions is only to help you in terms of giving you the not only the theoretical knowledge but also the experimental flavor as well. Okay, so Purandar Purnabrata Bag is asking about the star activity of a restriction enzyme. Uh, so you know that the restriction enzymes are of multiple types like type 1 restriction enzyme, type 2 restriction enzyme, and type 3 restriction enzymes. And mostly we are using the type 2 restriction enzyme because they are generating the palindromic sequences, and they are very specific where they are cutting actually, and because of that they are actually being chosen. But what happen is most of these enzymes, because they are very prone to degradation, so they are being supplied as the in the 50% glycerol. So when you add the restriction enzyme into your reactions, the final concentration of the glycerol should not be more than the 10%. So for example, in suppose you added the very high quantity of your restriction enzyme in the reactions, and the glycerol concentration gone up like 15% or 18% and so on. So because you have added the very high quantity of glycerol, that actually is going to affect the activity of these enzymes. So instead of cutting at their their definite restriction sites, they will start cutting at random places as well. So that aberrant restriction activity is called as the star activity. For example, if you talk about the EcoR1, the EcoR1 restriction site is GAATTC. So just after the G, it cuts, and that's how it actually generates the some overhang. But you can imagine that if I have a star activity, the EcoR1 will start cutting after G, but it also may cut at random places, and because of that it may actually will not going to generate the sticky ends. It will generate some random fragments, and those random fragments are not going to helpful in terms of the cloning also. So there are many ways in which you can actually avoid this star activity. I think we discussed in the course also, but like the one of the easiest way is that you ensure that the restriction enzymes should not have the more than 10% glycerol. So ideally we should have the 5 to 7% glycerol, and if you want to do that, probably you might have to take the restriction enzyme of a very high concentration, so that even 1 microliter of restriction enzyme should give you sufficient units, so that it should be good enough to cutting the your vector or the inserts. Apart from that, you can easily add some of the additives, like you can add the BSA and all other kinds of things, and that also is going to reduce the star activity. Uh, so I think Dr. Gautam is asking about the PDF material. So I see the PDF material is we have not provided, but I think it will be provided in due course. But if you really want to see the content, see this course is being the theoretical course. The content what you see is actually been taken from one of the my NPTEL course. So if you see the fundamentals of biotechnology, there is a course of fundamentals of biotechnology in the NPTEL course, which is actually a web course rather than a video course. And if you go through with the content of that particular course, you will see that whatever we have discussed theoretically, it is all available there, except that here we are incorporating some of the the lab sessions and all those kinds of things, and trying to make it more interactive rather than the web courses, which are only the theoretical courses. So in the meantime, until the content is not available, you can actually see the course which is called as the fundamentals of biotechnology, and I'm sure you will find everything there, whatever we are discussing in this course. So I think people like, for example, AU Korea is asking about the PhD admissions and all that. I think that is not something which is very much relevant to this course, so I'm not going to discuss about that. Uh, Then the Manjunath is asking, is it possible to make a bioluminescent plant? Uh, yeah, I think you can. If a plant is, if you can be able to overexpress the luciferase and all other kinds of enzymes into a plant, because if a bioluminescent animal is possible, like there are animals which are having these kinds of the proteins being expressed. Uh, so if that is possible, then why not the plant? But you might have to go through with the system, and then you have to also see if you prepare a bioluminescent plant, how it is actually going to interfere with its biological activity and metabolism also, because a plant is supposed to not emit the light when it is in the dark. So so because just like every other system, the plants also requires the photosynthesis. If you talk about the photosynthesis, the plant's photosynthetic machinery also require a recovery period uh, to regenerate the damages. If you don't know, but when there will be photosynthesis, the plant is capturing the sunlight, and that capturing of the sunlight is always being done by the carotenoids and other molecules, and then that captured energy is being transferred onto the photosystems. And once the photosystems are being taken up the light from the accessory pigments, it goes on to the high energy state, and that's how it comes down. But in this process, the photosystem itself get damaged, and majority of the plants actually utilizes the time in the night to recover from these damages, and that's how they actually synthesize the new photosystems, or sometimes they actually repair the damaged photosystems in the night. And that's how the plant, there is a day and night cycle for the plants also. If you keep a plant in light for a very very long time, and don't let them to go for the rest period, because if you generate a bioluminescent plant, the plant is actually going to emit the light, and that emit light is also going to be perceived by the plant to induce its photosynthesis also. So if you have a very very low level of photosynthetic activity, and you don't give the plant to go for a resting phase, and you know get ready for the next day, then I don't know uh, how how much it is actually going to interfere. um yeah, so I think preparing street lights with the bioluminescent producing plants is too much ambitious thing. It is very interesting to conceptually, but the amount of street light what you require, or the intensity of the light what you require, may or may not be possible. So that until you don't do these kind of experiments, it's very difficult to say. uh, if I if the only thing what you can imagine, if you have a bioluminescent plant, is it could be good for the ornamental purposes, but not for the purpose like it can be a source of light, because you cannot, if you, even if you have seen the firefly does not produce the bioluminescent continuously. It just produces the bioluminescent, it produces the light, and then it goes on off like that. So you can imagine, if you have a street light which goes on off, that may not be a very very comfortable situations. So bioluminescent plants could be good as far as the ornamental purposes is concerned. Like if you have a plant which glitters in the night, like if you have a plant in the park or something and it glitters, that may be good in terms of ornamental values, but not in terms of generating the source of light actually. So Atul is asking about whether the genetically modified culture for yogurt, etc., allowed in India. I think genetically modified organisms for consumption is not allowed in India. It's very strictly regulated, but very precisely, I really don't know actually the much in detail. But if it is a non-infectious and something like that, probably it could be possible. But uh, but yeah, exactly. I don't know if the genetically modified organisms or genetically modified bacteria is allowed for usage in preparation of the yogurts. So Dr. Gautam is asking about whether the assignment 0 is being considered for assessment. So assignment 0 is a mock assignment. It is actually being given only to give you the idea of what kind of assignments are going to come. So it is not going to be evaluated. Even I think if you have missed the assignments also, you don't have to be very much concerned about that, because out of the 12 assignments what we are giving, we are only going to evaluate the, or we are only going to take up the marks from the best 8 assignments. So even if you missed one or two assignments, you should not be very disappointed. You should still be continue, and that's how it is actually. You should be able to complete the course, and you will be able to get the certificates after completing the exam as well. So I think so that should not be an issue. And what Divya is asking, what will be the level of exam? So uh, the genetic engineering and biotechnology, this course, the level of exam is very high actually. It is going to be an objective type of questions, but the level is pretty, so in the exam, when you see, it is going to ask you the questions from the theoretical point of view. It is going to ask you the questions which are based on, mostly be on the like related from the lab course, like lab sessions what we have discussed, like the practical oriented questions. And then we also have the questions which are like of a reasoning based, or like where we probably give you hypothetical problems, and then you might have to solve or something like that. So it's pretty high level in terms of the like, if you if I give you a comparable exams, I think the gate is probably very closer to that. So whatever the questions you will see in the gate exam, like the biotechnology gate exam, except so if anything which comes from the genetic engineering, biotechnology, protein biochemistry or biochemistry portions, all that could actually be a content of this course. Uh, so Anvesha is asking about the stuffer fragment, by foreign DNA fragment we wish to insert, and why non-recombinant phages are inviable. Can you elaborate this question, because otherwise it's not very clear what you're asking actually. Okay, so Manoj is actually saying that he is doing some experiment with the wheat, and the wheat is when they are providing the light for 16 by 8, like means 16 hours light and 8 hours dark, it grows slow. And when it provides the 24-hour light, it's showing the normal growth as well as the spike, which factor is responsible. I really don't know which factor is responsible for the spike, but when you see when you s- I think with the wheat the situation is little different, because you know that the wheat is a monocot compared to the other plants which are dicot. So most majority of the plant what we use in our agriculture, like whether it is the except the pulses, like if you use the wheat or rice or all other kinds of staple food, uh, they all are belonging to the monocot. And the monocots are actually very good in terms of uh, performing the photosynthesis compared to the dicot, because the monocots are having a very high affinity for the carbon dioxide. And because of that, probably the reason that when you are doing a 24-hour light, it is showing a better growth compared to when it is showing the 16 by 8. So but the factors, I really don't know the factors, because it could be some of the factors like related to okay. So see, the issue is in a replacement vector, when the stuffer fragment get removed by the foreign DNA, it is actually leaves a viral genome. And because you have removed the stuffer fragments, that foreign DNA, that remaining viral genome cannot be cannot be circled and cannot be efficient enough actually to to produce the necessarily proteins so that it can be packed. Number one. Number two, the once you remove these particular stuffer fragments, they actually take up some of the essential genes along with that. And because of that, the remaining portion cannot actually packed and prepare the give you the virus particles. I'm sure it is clear now. So there will be no negative marking, Divya, in this exam, or so in the genetic engineering there will be no negative marking. All are like if you don't get correct marks, if you attempted wrong, I think there will be no negative marking. So I think you can. So Aditi, you can contact the NPTEL office and get the clarification about the hard copy of the certificate. To best of my knowledge, I think this time we are not providing the hard copy. Probably could be one of the reason could be that could be because of the COVID-19 pandemics. But the more detail, I think they will be able to tell you. And so I think I suggest you to contact the NPTEL office for that. And Anvesha, I think it might be clear now, right? So if you have any more questions, you can ask me. Purnabrata Bag, what is ADH? Can you elaborate what is ADH? Okay, ADP. Okay, so see whether it is ADP or whether it is the NAD plus, both are uh, the significant molecule uh, representing the low level of energy being present in the cell. So if there will be a short, if there will be a reduction in the ADP or the NAD plus. So what that mean is that the corresponding molecules are being produced in a very high quantity. So if you know that the cell is maintaining a very fine balance of the ATP versus ATP ratio, and similarly it is also maintaining a fine balance between the NADH and the NAD plus. So if there will be a reduction in the NAD plus, that actually will say that there will be a sufficient quantity of the NADH present. And if you have a very high quantity of NADH present in the cell, that means that the cell has sufficient energy for for maintain or for running its life cycle. So if you have sufficient energy, you are actually going to down regulate the metabolic activity, which means you are actually going to reduce the rate of the rate in through which the biomolecules are going to be burned down. Similarly, the same is the story for the ADP also. So, like if there will be a low level of ADP, which means you are actually going to have the high level of ATP. So, if you have a high level of ATP, there is no need to produce more ATP, right? So, if there will be enough quantity of ATP present, then why unnecessarily you are going to break down the glucose or even the lipids and all other molecules for energy production. So that's how the ADP or the NAD plus, if there will be an increase in the level of ADP and the NAD plus, it is negatively going to down regulate the Krebs cycles. I'm sure it is clear now. Uh, so Manoj is asking why when he is performing the experiment in his lab, his supervisor is always telling him that he should take the bacterial culture of zero point 0.6 to zero point 0.8. See, there is no reason of taking a bacterial culture less than zero point 0.6 OD, because if you see bacterial growth kinetics, once the bacteria crosses the zero point 0.6 value, it actually goes into the stationary phase, which means the zero point 0.6 to zero point 0.8 is the area which is actually the maximum amount of viable bacteria could be present in the your media, right? So once you have the enough quantity of bacteria, your production of the plasmid is going to be more. If you take up the bacteria, if you take up the cells from the log phase, the number of bacteria is going to be less, because still there is enough amount of nutrition available in the media, and the bacterial number can go up further. But because you have taken like 0.3 or 0.4, the amount of bacteria is going to be less. So that's how, if you go up to 0.6 to 0.8, you're going to have the sufficient quantity of bacteria, and the yield of the plasmid is going to be more. So Dr. Gautam, the final exam is going to be the MCQ based. So Anvesha, I think better you ask me what you could not understand when from the video, rather than I should explain you everything in detail, because the purpose of the live session is only to clear the doubt in the portion where you could not understand something. Because if I start explaining you the whole content, then I may miss the questions from the other people also. I really don't know, Yasha, whether there will be any plan of having a advanced course on this in the NPTEL, but I'm sure there are faculties who might be interested in giving the course on the advanced level as well. So I hope that you will get a course soon, because so Divya, I think this, if you go through with the content, I'm not saying that you will be able to qualify simply by going through with this course or content, but I'm sure if you go through with the content of this particular course, a larger chunk of the GATE exam syllabus is going to be covered. Apart from this course, what you need to read about is, you need to read about the bioprocess and the biochemical engineering. You might have to read about the immunology, and then very little you might have to read about the bioinformatics, because these are the another content what is been there in the GATE exam. So GATE biotechnology exam. So life sciences anyway is very different, but GATE biotechnology exam, if you are interested to attempt in 2021, then apart from this course, if you read about the biochemical engineering, bioinformatics and immunology, I think you will be in a very good position to attempt. and it may help you in getting the getting qualified also. So the format of this exam is going to be the multiple choice questions. Swagata is asking whether there will be genetic engineering and genome editing both are same. So genetic engineering is a generalized term for editing the genome itself or editing the organism itself. So genetic engineering actually involves the where you are actually going to uh, change or modify the organism either by putting an exogenous uh, 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 replicative units like plasmids or all those kind of thing, or you can actually genetically modify by simply integrating into genome. So genome editing is also a kind of genetic engineering only, because that also is going to allow you to produce the genetically modified organism. So both are not same, but both are so genome editing is also coming under the big umbrella of genetic engineering. So I think Yasha is asking about me something which is beyond the course or beyond the scope of this particular course. But since she asked me, and I think there's no question coming from the other students, I can easily answer. If you want to start up a basic laboratory of genetic engineering, and if you say what is the minimum thing what you require in terms of equipments, uh, so what you require is orbital shaker or the incubator, like the, so that you can be able to culture the bacteria as well as the you can be able to culture even. And if you are going to start the laboratory for manipulating the cells like mammalian cells, then you also require the CO2 incubator and as well as the biosafety cabinets. And apart from that, if you just want to do some genome like genetic engineering, you just want to produce the recombinant DNA and so on, then you start with the PCR, like you require a thermal cycler, you require a water bath, you require incubators, you require a laminar hood, and then you require a minor equipment like vertex, magnetic stirrer, and so on. So by these instruments you can be able to start up the labs, and then subsequently as the work will progress you can keep adding the other instrument as well. So I think I already answered about the Swagata's questions, that the genetic engineering and genome editing are same. The approach is different. That genome editing is where you are editing the genome, and but you still it's the same way of modifying the genome. So So Kami-sama is asking about how biotechnologists can be help in terms of the corona pandemic. So see, the biotechnologists are can help in terms of, and that's what I think the people are doing also, is they can actually be able to develop the better diagnostic kits. They can be able to develop the cheaper diagnostic kits. They can be able to develop the whether it's a RT-PCR based or the antibody based kits, which so that you can be able to detect the viruses. And the cost, if you reduce the cost, you it can actually help in terms of screening more and more samples, and that's how you can be able to track the complete profile, how the people are getting the infections. Apart from that, the biotechnologist can also help in terms of developing the vaccines, and subsequently, the biotechnologist can also help in terms of developing the drugs for this particular virus as well, because these are the aspects what we can be covered within the biotechnology. So biotechnology is a very big subject actually, where everything comes under the biotechnology, whether it is a drug discovery, diagnostics, or whether it is a vaccine development. So Sagata is saying that, sir, gene cloning part is not clear. So what part of the gene cloning is not clear to you? If you can elaborate, I think I will help you in to discuss that. वो लाइव सेशन का रिमाइंडर दे दो, लाइव सेशन वाला रिमाइंडर दे दो। क्वेश्चन हाँ So I think I really don't know about RNAs, but RNAs is everywhere. RNAs is everywhere in on this earth because RNAs is a very stable enzyme. RNA is there on the in the dust also. RNA is, but that RNAs cannot destroy the virus because the virus is not present in the form of RNA. The virus is present in the form of a particle, right? So if the virus does not come as a like a free flowing genome, right? So when you catch the virus, suppose you catch the virus from a surface, that surface is having not the RNA actually. It doesn't contain the coronavirus RNA. It contains the coronavirus particles. So if the particle is means the genome is actually going to be packed in the form of a particular, packed within the proteins, and they will not give any access even if there is a RNA present in your hand, because that is not going to inactivate the enzyme. The only way to inactivate the enzyme is that if you use the soap or the alcohol, because if you use the soap, the detergent what is present in the soap is actually going to denature the proteins, and that's how it is actually going to inactivate the enzyme, inactivate the virus, because the free flowing RNA is not going to be infectious, even whether it is from the coronavirus or whether it is from any other infectious organisms. Apart from that, if you use the alcohol also, alcohol is also going to do the same. It is actually going to dehydrate the virus, and at the end the enzyme, the proteins and all of the kind of machinery what is present within the virus is going to be inactivated, and that's how you can be able to kill the virus. You cannot kill the virus if you take the virus particle and treat it with the RNAs, because if you treat it with the RNAs will not get an access to the proteins. So and that's why you see the most of the time people don't use these kind of RNAs or DNAs or these kinds of enzymes. They always use the simple thing like the hand sanitizers or the soap, right? Because that's a way you can be able to get rid of the virus, and you can also inactivate and kill them. The third thing what people also do, like when they do the sanitization, they also throw, spraying the bleach as well. So bleach is also doing the same. Bleach is actually destroying the proteins as well as they are destroying the cellular machinery, like not the cellular machinery but the machinery of the virus, and that's how it is actually inactivating the virus. Okay, so once the code is disrupted, Kami-sama, once the code is disrupted, the virus is RNA is going to be present. Okay, and RNA is not infectious because the particles is infectious because it contains the proteins which allow them to attach to the cells, and that's how the cell is engulfing these virus particles, and that's how the virus is accessing the proteins like the mammalian cells. If you take the RNA virus, like if you take the RNA from the virus and you coat it onto your skin, nothing will happen because the RNA is not going to enter into the body, because RNA does not have any kind of receptors present onto the cell. RNA is going to be degraded automatically because RNA is going to be degraded by the RNases what are present inside the blood also. For example, our blood also contains different types of proteases, RNAs, DNases, and all that. So that kind of cellular protection machinery is already there in our body. So, but that not good enough for protecting us from the virus, because the virus is entering into the human body and then it is directly going into the lungs, and that's how it is actually going inside the cell actually. And by going inside the cell, it requires those coat proteins. So if you remove the coat proteins, or somehow if you destroy the coat proteins, then the virus is going to be inactivated, or it is actually not going to be infectious at all. Okay. 1 minute, 1 minute more. ये देखते हैं So I think I'll suggest everybody to ask me questions because our time is already getting over. So we have another 1 minute if you have any question to ask, and then after that we will wind up for the day, and then we are again going to see you soon. So that time you can ask me many more questions. So I think if you don't have any more question, I am going to wind up the today's live sessions, and I hope that you will like this course and you will understand the content more. And in our next subsequent lab sessions, we will going to discuss some more questions as well. Thank you.